Good morning, classmates, and good morning, Doc. Our report is about proteins. The presenters are yours truly and Sir Jason de la Fuente. Protein is the basic component of living cells and is made of carbon, hydrogen, oxygen, nitrogen, and one or more chains of amino acids. And there are three types of protein. They are fibrous, globular, and membrane. There are three classifications of protein based on its structure, composition, and functions. There are three types based on its structure: fibrous, globular, and intermediate proteins. There are two based on its composition, and these are simple and conjugated proteins. For its functions, there are three: structural enzymes, hormones, pigments, transport proteins, contractile proteins, storage proteins, and toxins. Proteins are polymers of the bifunctional monomer amino acids. There are 20 common naturally occurring amino acids, each contain an alpha carbon, an alpha amino group, and an alpha carboxylic acid group, and an alpha side chain or side group. These side chains are called R groups, which may be either nonpolar, polar, or uncharged, or charged, depending on the pH and the pKa of the ionizable group. Two other amino acids occasionally appear in proteins. One is selenocysteine, which is found in archaea, eubacteria, and animals, and just recently found is pyrrolysine, found in archaea. Proteins are polymers of the bifunctional monomer amino acids. There are 20 common naturally occurring amino acids, each contain an alpha carbon, an alpha amino group, and an alpha carboxylic acid group. Protein structures are organized into four levels: primary, secondary, tertiary, and the optional quaternary. The primary structure is the unique sequence of amino acids. The local folding of the polypeptide to form structures such as the alpha helix and beta pleated sheet constitutes the secondary structure. The overall three-dimensional structure is the tertiary structure. When two or more polypeptides combine to form the complete protein structure, the configuration is known as the quaternary structure of a protein. Protein shape and function are intricately linked. Any change in shape caused by changes in temperature or pH may lead to protein denaturation and a loss in function. The primary structure. The simplest level of protein structure, the primary structure, is simply the sequence of amino acids in a polypeptide chain. For example, the hormone insulin has two polypeptide chains, A and B. Shown in the diagram below, the insulin molecule shown here is cow insulin, although its structure is similar to that of human insulin. Each chain has its own set of amino acids assembled in a particular order. For instance, the sequence of A chain starts with glycine and at the N terminus and ends with asparagine at the C terminus, and is different from the sequence of the B chain. Characteristics of protein. Denaturing. It is a disruption in the ultimate function of protein when it is subjected to high temperature, acidity, solvents, and presence of some molecules. An example here is the frying of egg. The albumin curdles and it thickens when because it was exposed to high heat and oil. Another is strength. While different proteins have different properties in general, they can be extremely strong. This makes them ideal for structural elements in organism. Proteins provide structural elements in an organism such as muscles, bones, hair, and connective tissue to form a structure of the body. And this is what you can find here in your skin, the hands, the bone, and the collagen. The last two characteristics of protein are 6. It provides stored energy. Proteins can be metabolized by organisms for their stored energy. Last is the biological processes. Proteins are essential to life processes and functions because they form as the building blocks of the bodily functions. Here are the following updates on the best uses of protein in our world today. First is the protein dynamics research breakthrough could result in new cancer treatments, which was published by the Northumbria University. That researchers from this university in Newcastle and Bogazici University in Turkey have discovered a new way of examining the structure and dynamics of single protein molecules within the human body, which could help scientists better understand the progression and possible treatment of disease such as cancer. Next is the molecule attacks coronavirus in a novel way, which was conducted by University of Bonn in Germany. Scientists at the University of Bonn and the Caesars Research Center have isolated a molecule that might open new avenues in the fight against SARS coronavirus 2. The active ingredient binds to spike the protein that the virus uses to dock to the cells it infects. This prevents them from entering the respective cell, at least in the case of model viruses. It appears to do this by using a different mechanism than previously known inhibitors. The researchers therefore suspect that it may also help against viral mutations. The study will be published in the journal Anguante Chemie but is already available online. Third is the Eat Me, the Cell Signal of Death, conducted at Kyoto University in Japan. Scientists at the Institute of Integrated Cell Material Sciences and colleagues in Japan have revealed molecular mechanisms involved in eliminating unwanted cells in the body. A nuclear protein fragment released into the cytoplasm activates a plasma membrane protein to display a lipid on the cell surface, signaling other cells to get rid of it. The findings were published in the Journal of Molecular Cell. The fourth is the new technology enables ultra-fast identification of COVID-19 biomarkers conducted by the Universitatis Medicinae Berlin and the Francis Crick Institute. These two institutes have developed a mass spectrometry based technique capable of measuring samples containing thousands of proteins within just a few minutes. It is faster and cheaper than a conventional blood count. To demonstrate the technique's potential, the researchers used blood plasma collected from COVID 19 patients. Using the new technology, they identified 11 previously unknown proteins which are markers of disease severity. The work has been published in Nature Biotechnology. And that ends our report. Thank you.